purpose of this video is to compare water and methane and also discuss the transport in blood plasma. The properties of water have already been discussed in class. Methane is a waste product of anaerobic respiration in certain prokaryotes that live in habitats where oxygen is lacking. Methanogenic prokaryotes live in swamps and other wetlands and in the guts of animals, including termites, cattle, and sheep. They also live in waste dumps and are deliberately encouraged to produce methane in anaerobic digesters. Methane can be used as a fuel, but if allowed to escape in the atmosphere, it contributes to the greenhouse effect. Water and methane are both small molecules with atoms linked by single covalent bonds. However, water molecules are polar and can form hydrogen bonds, whereas methane molecules are nonpolar and do not form hydrogen bonds. As a result, their physical properties are very different. During this portion of the video, we are going to discuss some of the physical properties of methane and water. As was mentioned before, water and methane are both small molecules with single covalent bonds. The chemical formula of methane is CH4 and the molar mass is 16. On the other hand, water is H2O with a molar mass of 18. Methane has a density of 0.46 grams per centimeter cubed. Water has a density of 1 gram per centimeter cubed. In terms of specific heat capacity, methane is 2.2 joules per gram per degree of Celsius, compared with water that has a specific heat capacity of 4.2 joules per gram per degree Celsius. Methane's latent heat of vaporization is 760 joules per gram, compared to water, which has a latent heat of vaporization of 2,257 joules per gram. The melting point for methane is negative 182 degrees Celsius and a boiling point of negative 160 degrees Celsius. Water has a melting point of 0 degrees Celsius and a boiling point of 100 degrees Celsius. The purpose of this portion of the video is to discuss how different materials are transported in blood plasma, including glucose, amino acids, cholesterol, fat, oxygen, and sodium chloride, in their relation to their solubility in water. Blood transports a wide variety of substances using several methods to avoid possible problems and ensure that each substance is carried in large enough quantities for the body's needs. Sodium chloride is an ionic compound that is freely soluble in water, dissolving to form sodium ions and chloride ions, which are carried in the blood plasma. Amino acids have both negative and positive charges. Because of this, they are soluble in water, but their solubility varies depending on the R group, some of which are hydrophilic while others are hydrophobic. All amino acids are soluble enough to be carried in blood plasma. Glucose is a polar molecule. It is freely soluble in water and is carried dissolved in blood plasma. Oxygen is a nonpolar molecule. Because of the small size of the molecule, it dissolves in water, but only sparingly, and water becomes saturated with oxygen at relatively low concentrations. Also, as the temperature of water rises, the solubility of oxygen decreases. So blood plasma at 37 degrees Celsius can hold much less dissolved oxygen than water at 20 degrees Celsius or lower. The amount of oxygen that blood plasma can transport around the body is far too little to provide for aerobic cell respiration. This problem is overcome by the use of hemoglobin in red blood cells. Hemoglobin has binding sites for oxygen and greatly increases the capacity of the blood for oxygen transport. Fat molecules are entirely nonpolar, are larger than oxygen, and are insoluble in water. They are carried in blood inside lipoprotein complexes. These are groups of molecules with a single layer of phospholipids on the outside and fats on the inside. The hydrophilic phosphate heads of the phospholipids face outward and are in contact with water in the blood plasma. The hydrophobic hydrocarbon tails face inwards and are in contact with the fats. There are also proteins in the phospholipid monolayer, hence the name lipoprotein. Cholesterol molecules are hydrophobic. Apart from a small hydrophilic region at one end, this is not enough to make cholesterol dissolve in water, and instead is transported with fats in lipoprotein complexes. The cholesterol molecules are positioned in the phospholipid monolayers with hydrophilic region facing outward in the region with the hydrophilic region facing outwards in the region with the phosphate heads of the phospholipids.